And we move on to our next speaker, Aya Kigel from Yariv's lab.
Hi everyone, my name is Aya and I'm from Yariv Wine Lab, and today I'm gonna present you my PhD, which is the molecular landscape of B-cell immunity at the maternal-infant interface. So neonates are exposed to infection during early infancy due to their immature adaptive immune system. So during pregnancy, maternal antibodies transfer to the fetus via the placenta, and later, after labor, these maternal antibodies are consumed by the infant. Both of these transmission routes can be considered as a double protection layer that the mother provides her offspring during early infancy. Despite the importance of these transmission routes, little is known regarding the molecular features of antibodies and B cells that are part of this mechanism. The recent reintroduction of the maternal immunization became a routine strategy intervention that health authorities recommend. The maternal immunization enhances the transfer of vaccine-specific antibodies to the fetus and later through breast milk. So today I'm gonna talk about the second layer of protection, which is the breast milk. So breast milk is a complex fluid that evolves over time. It starts with the colostrum, then the transitional milk, and then it evolves to mature milk. It contains a lot of substances, including water, vitamins, mineral, and of course, antibodies. So breast milk includes 3 different isotypes, IgM, IgG, and IgA. And IgA is the most dominant one in breast milk. The accelerated COVID-19 vaccination campaign initiated in Israel in December 2020, but the vaccination campaign didn't include pregnant and lactating women because they were excluded from clinical trials. So it was an urgent manner to generate data regarding the vaccination of this population, and we were happy to be one of the group that generated information regarding the vaccination of this population, and to help health authorities to make a concrete recommendation regarding the vaccination. So at the start, we asked ourselves 2 important questions. What is the difference between the dynamics of the antibody response in breast milk in comparison to the blood, and what is the neutralization capacity of these breast milk antibodies? So we collaborated with Lis Maternity Hospital in Kigel, and they were able to recruit women that got the vaccine during their lactation. So the cohort comprises of 10 lactating women that received 2 doses of the COVID-19 mRNA vaccine at the interval of 21 days, and we collected blood and breast milk samples in 4 time points, 7 and 14 days after each dose. So the first thing we saw that the response in blood and breast milk was synchronized between the 2 compartments in IgG and in IgA also, and it peaked 7 days after the second dose of the vaccine. And the next thing we saw that all the 10 lactating women show neutralization capacities, and especially the IgG isotype. IgA was also neutralizing, but a bit less than IgG. The next thing we asked ourselves is we wanted to evaluate the antibody response in different isotypes at different lactation stages and determine the optimal vaccination during pregnancy this time. So we collaborated with Dana Dweck Children's Hospital, also in Kigel, and they were able to collect us samples from 62 women that received the vaccine during their pregnancy. they received the vaccine during their second or third trimester, and 3 different samples from 3 different lactation stages was collected: colostrum, transitional, and mature. So the first thing we demonstrated is the existence of the vaccine-specific antibodies in all lactation stages. And we showed that the highest concentration of antibodies was seen in colostrum, and then between the transitional and the mature milk, there was not a significant change in the concentration. The next thing we wanted to see is when is the best time to get the vaccine, second or third trimester. So if we're looking at IgG isotype, we can see that it's better to get the vaccine during the third trimester because the concentration of IgG were higher. But if looking on IgA isotype, we can see the opposite. So it's better to get the vaccine during a second trimester. Given the crucial role of IgA and the higher concentration of this isotype, we recommended that women will receive the vaccine during their second trimester. So a great focus has been dedicated to antibody in breast milk, but immune cells are also present in the human milk. So it includes neutrophil, macrophage, lymphocyte. The majority of lymphocytes are T cells, but there are also 2% of B cells in the milk. So what is the role of these B cells? Where did they came from? What is their phenotype? How they function? So to start and answering this question, we came back to our previous cohort, and we were given the opportunity to collect B cells from breast milk and from blood samples. And for the first time, analyze these B cells, and we were surprised there is no data regarding these B cells at all. How I go back? So to start and understand what measures we analyze, I need to give you a short introduction regarding 2 major diversification mechanisms that B cells undergo. So the first mechanism is the VDJ chromosomal rearrangement. It occurs in the bone marrow, and it's antigen independent. So V gene segments are recombined with D and J segments in the heavy chain, and they creating a diverse population of B cells. These B cells can now migrate to secondary lymphoid organs, and these naive B cells can encounter with an antigen, and they start the affinity maturation process, which include the proliferation and clonal expansion, then the somatic hypermutation that improves the affinity of these B cells towards the antigen, and then the clonal selection, which means that B cells that produce better antibodies are kept, and B cells that with disadvantaged mutation undergo apoptosis. In the end, the affinity maturated B cells can differentiate into memory B cells or plasmablast and migrate via the blood to other tissues and organs. So if I'm going back to the blood and the breast milk B cells we collected, we isolated vaccine-specific B cells from each compartment, memory B cells and plasmablast, and sent it to a BCR-seq. The BCR-seq clones we divided into 2 groups. The first group was the BCR common clones, which means it's shared between the 2 compartments. It's clone that we have found in the blood and in the human milk. And the second group is the uncommon clone, which means there are clones that are unique to each compartment. So the first thing we measured was the isotype distribution, and we saw that common clones between blood and milk, so in the milk, they expand toward IgA and IgM isotypes. Moreover, they show a higher somatic hypermutation rate in the milk. So another thing we saw that surprisingly, not just the antibody level was synchronized between the two compartments, but also the transcript level of the common clones between the two compartments were also highly synchronized. So if I want to conclude, we showed that vaccine-specific antibody response following the mRNA vaccine is highly synchronized between the two compartments. Neutralization capacity of breast milk was evident at all women who participated in the study. Maternal immunization with the mRNA vaccine during second trimester of pregnancy provide higher concentration of IgA throughout all lactation stages. And for the first time, we sorted, isolated, and performed BCR-seq on breast milk residing B cells. We found common clones between the two compartments, and the antibody repertoire signature suggests that B cells migrate from blood to breast milk and continually evolve in breast milk tissue. I want to thank all our collaborators and funders, and of course my lab members, and thank you for listening. Thank you.
So did you profile the B cells that are in the breast milk in terms of subpopulations? Do you know if these are naive, plasmablasts?
Yes. So we had 2 time points. So 7 days after the second dose, we saw more plasmablasts. And 14 days after the second dose, we analyzed the memory B cells. So we're collecting also plasmablasts and also the memory.
Have you seen differences in the composition of B cell populations in the blood compartment versus the breast milk?
Yes, because this is initial results. We are working on it as we speak, and the analysis on the plasmablast is, I'm in the middle of it. But yeah, we saw a different, like in the isotype usage and sub-isotype usage also. In the milk, it's more a secretory isotype, and in the blood, it's more IgG. Also, the somatic hypermutation was higher in IgG isotype in the blood. Yes, if we're comparing this specific isotype.
I have a quick question. Do you have a mechanistic explanation for the difference, for the fact that there's higher IgA in second trimester but higher IgG in third trimester?
No, but we try to answer it also, but in other research that people do on other maternal immunization, they've seen this same, yeah, that sometimes or sooner in the third trimester or even the second trimester, higher concentration. But this field is understudied, so
Yeah. More work for you.
yes.
Great. Okay, thanks, Aya.